Thank you. What I really want to talk about is kind of how one can try to meld questions in evolutionary biology with those of kind of more molecular and cellular biology in the context of using bacteria in the lab, and how by doing evolution in the laboratory environment, you have kind of a stage in which kind of all things happen, new mutations which affect the proteins, which affect how the cells work, which affect their fitness and evolutionary dynamics, as well as sometimes new ecological dynamics arise. And so kind of everything, all of biology goes on within a completely artificial environment. So it's absolutely and totally natural selection in terms of this stuff that I'll be talking about, but of course the environment is completely under our control and artificial, flasks or little 96 well plates, etc. And so I would say much of biology, kind of the heart of biomedical sciences is kind of looking. I have no pointer. Does there happen to? I'll use good old school technology pointer. So, and this I suppose is also a good disciplinary item too. If there's talking in class, right? I'm sure many of you wish you could use such a thing often, perhaps. Much of biology that I think most people learn about tends to be about kind of determining what makes organisms the way that they are in terms of phenotype, in terms of how does changes in the genotype, various mutations, etc., changes in the environment, etc., affect phenotype, whether that phenotype is health or the development of an organism or the ability to grow on some compound, etc. And so for many people, we tend to think, for biomedical sciences, the model organism is something like this. I realized after I drew the picture that it's probably, the self-envisionment of, a professor, given the relative ratio of body parts, etc. In this talk, my model organism is something a little bit more like this. And so I'll be talking about microbes, but there's no reason why it has to be. Hopefully, I'll be able to make the point in terms of why it's nice and why, given the difficulty of the questions, starting with organisms that are as simple, and in some ways they are, certainly advantageous. And so another kind of thrust in terms of biology has come more from the side of naturalists, etc., and ecology and evolution. And the questions there tend to be somewhat different. They tend to look at how do our various phenotypes end up being selected in the environment, how do you have ecological interactions between different organisms, how the environment affects the organism and they affect their environment, how by having different fitness, which fitness can be defined as relative reproductive success, ends up leading to population dynamics that change the ratios of various genotypes, how mutations come in and affect those genotypes, etc. And as much as we're very interested in how things end up affecting the genotype and how phenotype gets selected upon, there's often relatively little guts to kind of the discussion in between. And so it's kind of two parts of the puzzle. And so what I'd like to suggest is that it's important to try, where possible, to consider kind of the entire picture all at once and try to really find ways in which one can connect meaningfully from the genotype through physiological functions to the phenotype, through ecological and evolutionary functions to fitness and how it changes the genotypes. And so there's, these days, there's a great tendency to come up with many, many new phrases, the transcriptome, the genome, the proteome, the everything-ome, etc. And so I think, a novel term perhaps for something like this would be biology. And really, this is the whole picture, so as to say. It includes all of the above, how things work at the level of molecules and cells all the way up to organisms and populations and ecosystems. And so, like I said, I'm going to tell you about how we try to use the simplest sorts of ecosystems, these laboratory populations, often with a single microbe in them, to try to understand across these various scales. And so I had given a talk at the NIH just a little while ago that I used for these slides, and I believe this is microbes and infectious disease, is something like that as a title. And so I think one case in which it's very clear where kind of evolution affects us on a very rapid timeline is when you do consider infectious disease. Whether you're thinking about like HIV, and this is for example the phylogenetic tree of HIV strains isolated from a patient through time, where you're talking about the evolution of antibiotic resistance, or even cancer. I mean, cancer really, even though it's something that's deleterious from the point of view of the organism, from the point of view of the cells, the mutations to oncogenes or tumor suppressors are essentially beneficial mutations. They're allowing them to outgrow their neighbors. And so the process of cancer growing up, and beginning to form a group of faster growing cells, it really is very similar to the sorts of processes that we look at in a flask. And it's not to say that, of course, evolution has set the stage in terms of kind of the host genotype, us or whatever host that you're thinking of, et cetera. But these are, of course, things that happen on a longer time scale and often a little bit harder to kind of wrap one's head around. And I think one place where it's really kind of critical and very, where evolution can affect you, incredibly rapidly is when you're thinking about infectious disease. So I'm going to ask kind of an unusual and I think kind of ridiculous question, and this is one of the kind of the hearts of the work that we're trying to do, and I think it's something that's in some ways almost impossible, but I think it's really important, and I think since intermediate steps along the way are valuable, I think it's worth kind of trying to start off one's career with kind of a ridiculous question. And so mine that kind of latches onto my varied interests is, can adaptation be predicted? And so, kind of going back, would I have been able to what extent could I have actually, predict mutations, along the way here? And I mean, so you can think about, predicting adaptation in many different ways. You can ask, will it happen or will it not happen? If you have a small enough population, as you probably, like cheetahs, if you have a small enough population, there may not be enough either genetic variation or factors such as drift and just random sampling in the population may make either neutral or alleles rise in number. And so, you get this issue when populations get too small, they can actually start to go down in fitness. So adaptation is not always a given, but assuming that it is a large enough population and you have the potential genetic variation that would give you more fit forms, sure, the answer would probably be yes. You could also ask, kind of how fast does adaptation occur? What's the rate at which fitness would increase through time, and how does that change through time? But what I'm talking about here is something a little bit different. So I think kind of a way of visualizing it is thinking in terms of, I'm interested in trying to actually be able to predict the relative likelihood of various targets being the site at which beneficial mutations occur. And so if you think about, lining up all the genes in the genome and ask, what's the probability of the first beneficial mutation arising in gene A, B, C, et cetera. And so whether you're thinking about, this could be a tumor cell, and you're lining up, all the oncogenes and tumor suppressors, and given a particular cell and a particular person's genotype, et cetera, can you say something about what's the relative probability of what's likely to happen, given what you have now, next? And then once that's occurred, how does that change the picture? So once a particular mutation has been incorporated, how does that now affect the distribution of the probabilities of the next thing occurring? Right? So anybody agree that this is a fairly crazy thought? I mean, this is really this to some extent would really be forcing one to really know everything across kind of that diagram of biology in terms of how cells work, as well as evolutionary processes that act upon populations of cells and organisms. And so, all right, this is somewhat crazy, I think. It is rather difficult. I think there are sometimes trivial cases that help illustrate how this can be possible. And one is antibiotic resistance in cases where you're simply having to change the target of the antibiotic. If the antibiotic attacks RNA polymerase or attacks the ribosome, this is actually not so hard to predict. Guess what gene is going to have to change to avoid the antibiotic resistance? That particular, whatever your gene is that is the target site for the antibiotic. So in some cases, this is going to be trivial. Essentially all of the probability is a single target. Great, but that's not particularly useful. You can imagine the opposite scenario. What if everything was kind of equally controlled? Like, nothing really matters all that much. Everything matters just a little tiny bit. So you could get a worst case scenario where things were completely random. And you'd have just kind of a flat distribution of however many genes there are, and all of them share some really, really, really small percentage chance of being the next thing. And thus what you'd see in evolution would be just complete random chance, just whatever the dice roll. And in terms of what's more mutationally likely would suddenly shape much more kind of what you see. And so the reality is probably somewhere in between here for most cases. It's not going to always be as easy as just one gene. It's not going to always be just simply flat. It's likely there are going to be certain aspects, certain features, and so if we knew how a system functioned, maybe we can begin to understand this. And I think this distribution to some extent is knowable, at least in aspects about it and in certain situations. And I think it's important. If you're thinking about cancer, for example, if you know that you have some polyp that's beginning to grow and it has these three oncogenes or tumor suppressors that have already been hit, what information does that tell you in terms of what's the next likely set of targets given what has already occurred? And these can be either things that you're watching out for, trying to detect mutations in those, or maybe even things that you can prevent. If you know they're likely, after having taken these steps, to make this next step is highly likely, you can try to specifically target that so that that's not favored as the next step. And so in terms of what would be needed to predict adaptation, I think, so this is kind of a four-step program. There are kind of four aspects that are needed, I think, in terms of being able to understand this. The first is kind of this question: how much fitness increase could you get per how much you can change in the system's components? Essentially, kind of how much bang for your buck could you get in terms of fitness by being able to change different parts? Whether you're talking about a cancer cell, if you could turn up this gene, that gene, if you could do anything, which of them is going to make the cell grow fastest? So if it's my bacteria in the flask, if I could change any given enzyme, et cetera, by some amount, what points in the system are the most sensitive? So this requires kind of quantitative system level knowledge to try to be able to make this sort of prediction. But that's if I could do anything. Of course, since the changes are coming through mutation, there's the second aspect in terms of what's the actual mutational distribution at all of these different loci. What's the relative rates at which you can mutate these various targets, and what's the kind of the distribution. Some enzymes, it may be very hard to make it any more than like 5% more active than it is right now. Others, it may be very easy to change its activity by great amounts. And so this would be the other important aspect. So if we knew how much you could change any of the parts and how much the parts affect the system, then we know how much can mutations affect the system. Kind of what's the total distribution of all the potential changes in fitness that are possible. And you'd think you'd be done, except there's still space at the end of this. And so that's everything that could be done. The question is, what actually needs to be done? Because and so this is where the kind of the population biology part of that early slide comes in. How large of a population actually are you? Kind of sets how good do you need to be to actually be able to win. And so one way of thinking about this, my student of mine came up with for his qualifying exam in this area is you can ask like how good of a basketball player do you need to be to make your high school team? Well, you're sampling across the high school, and so you need to be one of the top 12 players in the high school, or top 5 if you want to be a starter, kind of thing. And so you can that's going to take some degree of skill that probably surpasses most of us in this room. But then you can ask if you go to college, if you go to the NBA, that's probably not good enough because there are lots of other people who are that good, if not better. And so as you move through population sizes, kind of how good you even need to be the winner matters quite a lot because in the case of basketball, there are only 5 starting spots. In the case of adaptation, ultimately only one lineage will have gone to fixation. And so as population sizes get larger and larger, what it takes to actually be the winner goes up and up and up because you're sampling from a much larger pool. And then finally, there's one more thing, which is that as systems begin to change, all of these aspects here also change, such that once you begin to make mutations that have changed how your system's functioning, all the distribution and maybe even some of the rates in terms of what's possible to change also can change. And so these interactions between genetic loci, these are called, this is called epistasis. I'll mention it again at the end, assuming I get there. But so basically, once you begin to make moves and you begin to incorporate beneficial mutations, what becomes possible, and if you make this particular change to these particular enzymes, it may throw some other aspects out of whack, and so that new things become beneficial that would not have been had you take a different first step. And so, if you could know all these various aspects, you'd have the chance to try to actually predict adaptation before it happens, and this would really be kind of synthesizing kind of understanding of biological systems as well as systems of populations. And so I'm going to talk about metabolism, and so it has kind of a particular language in terms of talking about these various parts, and so in terms of thinking about how the genotype affects a metabolic system, it affects the levels of the properties of enzymes, whether it be how fast they work, how well they bind to their substrates, etc. And then these changes affect the flux, the flow of molecules through metabolic pathways. And the flow of molecules through particular pathways, as well as sometimes the concentrations themselves, this is what determines kind of cell-level phenotypes such as growth rate. The goal of a bacterium is to make 2 bacterium. It needs to throw its food into biomass, and that depends upon how fast that it can actually do so. And so this is kind of going to be, this could be any sort of system, but metabolism I think has some nice advantages because it's something that can be quantified in terms of thinking about how fitness, in a simple system at least, like ours, fitness, as I'll talk about, largely relates to growth rate. And growth rate is largely dependent upon the flux of metabolites. These are, which are dependent upon enzyme properties, and all of these things are things that you can measure, you can quantify them. You can even, as I'll talk about, you can use models to describe how such systems work. So I think it's relative to say, how does one cell bud and make 2 cells? That sort of kind of physical process of one cell becoming 2 is much more difficult to kind of quantify at this stage. Not to say that it won't be ultimately, but I think metabolism is a good place to start. And so, in terms of taking these questions, we can ask, kind of how strong does selection act upon different enzymes? What's the distribution of effects, mutations that affect these enzymes? How large of a selective effect does it take to win? And then how do mutations to different enzymes kind of affect each other when they begin to interact with each other? So these are kind of the broader sets of questions that we're trying to address. And like I said, this is a talk about microbiology, and we do study a microbe. And so our bug is Methylobacterium. And so this is a leaf print. And so if you take a plate that contains methanol as its sole carbon source, this bug happens to be able to eat methanol. And this is kind of fading out on me. There we go. And so you can stamp it into a plate, and you can get these really nice kind of constellation of pink colonies. Most of these bugs in this genus tend to be pink colored due to carotenoids. And so my bug, Methylobacterium, it's a plant epiphyte. It lives on the surface of leaves. And as I just mentioned, it can grow on things like methanol, on single carbon compounds. And so although, I doubt at least that anybody had ever heard of Methylobacterium before you came in the room, and that's okay, I'm not hurt. It has been a model system, just not as much as E. coli, for example. For the last 50 years, actually, this bug's been worked on in terms of trying to understand just how does it do this in terms of being able to eat things like methanol. And the reason why something like methanol is unusual is that growth on something like methanol or other C1 compounds, these get oxidized to formaldehyde, and then the formaldehyde gets further oxidized on through to CO2, and at various oxidation levels gets assimilated into biomass. And so every single carbon while you're eating goes through formaldehyde, and it doesn't take much to realize that this poses an issue. I mentioned, especially like in our laboratory conditions at least, fast growth is what's being selected upon. And so you want the flow, you want these arrows to be as large as possible. But of course, if you let kind of the concentration, the pool of formaldehyde build up, the cells are going to pickle themselves in no time flat. And so they clearly have to have a well-controlled, well-regulated system to be able to handle doing this. And so I actually spent my time in graduate school understanding kind of the physiological aspects, just how is it that they deal with the formaldehyde that they make during growth on methanol. And then became interested in evolutionary questions, and kind of the questions, as you'll see, kind of stemmed from my particular bug, and so I kind of brought my system with me onto my postdoc and now to my lab. And so this looks nice and simple. I've drawn it with just a few arrows. To be able to do this requires about 100 genes that are required for single carbon growth. And these genes have very clearly been acquired through horizontal transfer, which is a process I'll talk about actually in just a minute. I think it's one of the critical things to talk about in terms of microbes. The other thing that's kind of interesting about these bugs is that almost all organisms, all bacteria that can eat things like methanol, methylamine, methane gas, etc., are very, very highly specialized. That for many of them, that's all they can eat. In this case, this bug can at least eat a few other compounds, but it's lost the ability to eat all sugars, a lot of the things that are kind of typical. And the interesting thing, too, is that you don't just find all these C1 utilizing bugs related to each other. They're sprinkled all over the phylogenetic tree. Genes involved, these 100 genes, they were not reinvented time and time and time and time again. they've been passed around, as I'll talk about, through horizontal gene transfer. So you have a case where many separate times, organisms that were not able to eat C1 compounds acquired the genes to be able to do so, became reasonable enough in terms of doing it that they're still doing it now, and almost every single case have either completely lost or almost completely lost the ability to eat anything else. So it's a really interesting case kind of repeatedly seeing specialization arise after the incorporation of a new pathway. And in order to kind of better understand some of these bugs, I'm also, besides kind of this experimental work, I'm leading a group where we're analyzing the genome sequence of 6 more methylobacterium strains. We'll end up having a total of 8. And so it opens up kind of complementary approaches to thinking about these bugs besides what we do in the lab. But so I wanted to step back for a minute and talk about horizontal gene transfer. You know, in terms of thinking about microbes versus macrobes, what really matters? Like, why, when we talk about evolution, for example, what does it really matter whether we're talking about zebras or, Staphylococcus? And I think that there are a number of important ways that they are different, and there are ways that they're relatively similar. Any thoughts? Why we should care?
They're both living.
They are definitely both living, yes. So luckily that is true.
Generational time is a lot different.
Yeah.
At least from a research perspective.
Yes, exactly. This is why I do not have a herd of zebras. Yes. This is usually true. I will say that there are microbes in the deep sediment, ice cores, etc. They probably double every decade. There are some that are certainly very, very growing. Any other thoughts? Yeah, so I think this is a big one. Now, there are plenty of macrobes. For example, a lot of plants that self, and so that some, or just simply grow vegetatively. You can, you grow up some plant, and you take clippings and you give them to your friends, etc. And I mean this happens in nature too. So it's not to say that all reproduction of macrobes happens sexually, but obviously most of, in terms of what we think about, does. And so we tend to think about microbes as being asexual, and it's certainly true that reproduction and sexuality are separate, which tends not to be true for larger organisms. But as we'll talk about, the bacteria I think are best thought of as being kind of parasexual. They do have a degree of sexuality. The other I would say that's different in terms of thinking about this whole high school basketball player to the NBA kind of analogy is just typically huge population sizes. But even that isn't necessarily always true, because sometimes if infections are only seeded by one or a few microbes, effectively the population size becomes very small, because then any change that was in that one microbe goes to fixation in that one individual, even though it was just kind of randomly drawn from what was there before. And so you still can, not something microbes do, but many at least also have very large population sizes, which also plays into kind of some of evolutionary dynamics. So like I said, I would say that they're kind of parasexual, and so there are different ways that genes can be exchanged. Anybody know those?
Yeah, so can they exchange genes with members of different species?
They can, yeah. And so and what's interesting is there's kind of 3 different ways in which they can do this.
Transduction.
Yeah.
Conjugation.
Awesome.
Transformation.
And so yeah, so these are kind of generically the three ways in which DNA can be transferred around. And this is actually important even to the research that I'm going to talk about, because the system that we're evolving is one that actually contains an engineered horizontal gene transfer, as you'll see. But so one way, a way that probably, if anybody does any sort of kind of lab may have used, is using transformation, get competent cells and try to make them take up DNA, take up plasmids. And so transformation is the uptake of naked DNA. And so some cells you have to just beat them very silly to be able to take up any DNA. And so with E. coli, you go through this crazy heat shock process, and you had to make kind of special cells with calcium chloride and blah, blah, blah. Or sometimes you electroporate them, where you have to get like all the salts out, and then you basically put them into an electric chair and zap them, and it sucks the DNA through the membranes, et cetera. And so some bugs, you really have to force this issue. Other bugs actually are naturally able to do transformation at very high rates where they can specifically bind DNA and bring in DNA fairly often. And so on one hand, this sort of uptake of DNA, this has a potential to bring in the new genetic information, but DNA, of course, is also food. And so the bugs that can do this, that are naturally good at doing this, it's unclear to what extent the selective pressure has been simply to eat something that's very useful, DNA, versus being able to take in new genes, or to some extent both. So that's one. So another is transduction, and so this is infection-mediated transfer. And so there are various viruses that, when they package their own DNA, can package some of the host DNA. And now it's in a nice, it can accidentally get packaged. It's in a nice, protein shell that can infect the next cell. And if it gets recombined in, then it has a chance to end up in the genome and perhaps end up doing something for the organism. And then the last is conjugation. And conjugation is where usually this happens by plasmids, which are like small circular chromosomes that are have to some extent their own life. And many of them have the ability to jump ship and move to new cells. And they encode all of the necessary machinery to replicate their DNA and to make this pilus that attaches onto the other cells and transfers the DNA in. So it's direct cell-to-cell transfer. So these are the three various ways in which DNA can be transferred. And they certainly all matter to different extents, in different environments and for different taxa, et cetera. And the other thing I wanted to mention about horizontal transfer, so in terms of thinking about exchanging genes, bless you, I think there's really two types of transfer that are important to think about. One is totally new DNA. So in terms of this idea of like genes moving across species. So of course, everyone's probably heard about antibiotic resistance and how it can move across various species, some that are commensal, happy bugs that live in us, and then a pathogen comes in, gets those genes, et cetera. And so in terms of, sometimes these can be, like I said, just genes that just simply were not there at all, and can become, if they end up becoming incorporated into the chromosome, you end up having this region, and as you see that it's clustered, where you've got totally new genes that were not there before. And many suites of genes that allow, like say, E. coli to be a pathogen, like the Jack-in-the-Box E. coli versus normal E. coli, came in large 50 kb big chunks of DNA through mechanisms like this. But the others, you can also exchange alleles. So this is where you've got some bit of DNA plops in, and suddenly you've got something that wasn't there at all, moves into the chromosome. Exchange of alleles, where you have some allele that has some point mutational difference, and then you've got the rest of the chromosome that has the same region too, and you can get recombination, and now the chromosome has the mutation. And so this is much more like normal sex. I mean, when we talk about sexual recombination, we get one allele from our dad and one allele from our mom for any given gene. We don't get a totally new set of genes from mom or a totally new set of genes from dad, no matter how much that might seem like that's true. And so this is a much more a typical sort of in terms of the way we think about sexuality. And this is something, this exchange of alleles is more so to say within species. Any ideas why it has to be within species?
Don't they have to form a, it's pylon, I guess, but does that have to join the two?
So that, believe it or not, that can happen across, so a lot of plant biotechnology actually has come from the fact that agrobacterium, a particular bug not so far away from methylobacterium, can make pili with plants. And you can also inject into like C. elegans, into yeast, into all sorts of things. And so the ability to exchange actually is, it's remarkable. I find it surprising, but for at least many types is very, very broad. But there's another aspect here, and it's because in order to get homologous recombination, that the DNA has to be similar enough to each other for those rates to be able to occur at something appreciable. And so this process drops off very quickly, exponentially with sequence distance. And so by the time you're at like even 98% similarity between two genes, it's already a couple orders of magnitude less frequent. So this is something that only can kind of occur within species, whereas totally new DNA, we have cases of DNA from like bacteria and archaea that have been swapped back and forth. And of course, these are two groups of microbes that share a common ancestor, oh, I don't know, 3 billion years ago, something like that. And so the first process certainly has no absolute limit in terms of what DNA can be incorporated, but the second one is something that's of close relatives. And so just very briefly in terms of how do we know something was actually a horizontal gene transfer, one typical way is if you look at a phylogenetic tree, you can find genes that just kind of have a pattern that doesn't make any sense. And so if you have like some gene that's over in this organism here and in these three here. And so the thing is, anytime you see this sort of pattern, there's always multiple explanations. So one could be that you had a horizontal transfer going one way or the other, and depending on how many times you sample on each side, you can make an argument, or maybe you have some other reason to make an argument which way it went. It always could be explained by selective loss, that everybody, wrong pointer, that the gene actually was present in the common ancestor of everything. It was kept here and was lost in absolutely every other lineage except for kind of the one that you found it in again. And so in the end, we don't know. But so one ends up making an argument of parsimony, that the simplest explanation would be the one that requires the least number of kind of moves. But of course, that's not to say that loss is equal to horizontal transfer. You might have to kind of weight how many losses per horizontal transfer you think is reasonable based on data. So that's one way. The other way is that you often find kind of signatures in the DNA that look unusual. So the genes look foreign. Besides kind of just, so that I guess was the genes look foreign too, phylogenetically. They look foreign because nobody else around you has them. But so how could a gene look foreign?
Have to do with like different GC.
Exactly. You can get cases, so even though, because the code is degenerate, there are many different ways for most amino acid residues to encode them. And so often it could be a GGA or a GGC or a GGT or a GDG that encode glycine, for example. And so different bugs use these at different frequencies. And so sometimes it shows up just simply at the level of GC percentage. And so most of the chromosome in this particular bug was about 50%, but then there's this region, island for about 45 kb, where consistently the GC percentage was much, much lower. It just came from a bug that had a different mutational bias, tended to be towards A's and T's, and then pops back up. And thus you also tend to see things that are kind of clustered. There's often regions where, kind of common regions where things can integrate in. So genes that are shared or very closely the same across many bugs are sites where you very often find these. So like tRNAs are something that all organisms have, and they're fairly well conserved. So these can be recombination sites for totally new things to get incorporated, et cetera. So you get these like islands where you look at this and you say, all right, this just is not possible by chance to get this many genes in a row. Suddenly, the skew, the DNA just looks totally strange. And so the nice thing is you can actually use this, look across an entire genome, find all the genes that look foreign, and then even try to estimate, given kind of the rate of time, if something came in looking like this, you can ask kind of how long would it take to look like that. What's the rate of change in which it kind of slowly, because most of this is a neutral process. It's not necessarily selected upon. It's just there's different mutational bias. And then you can estimate kind of how long have genes, those that are foreign, how long have they probably been there. And so somebody did this with E. coli. And the weird thing, it looked like all new genes that came in just yesterday, that on a relative kind of time scale. And very, very few had been there for a longer period of time. This is age in the genome in millions of years. Were like essentially now, some in the 1 to 10, and then very few in kind of more than that, millions of years. So the thing is, E. coli's lifestyle probably hasn't changed. What this probably hints at is many things are constantly coming in, and they're also constantly being lost. And so horizontal transfer, just like any type of mutation, it's not always good. It's just simply DNA coming in. It's just variation. And so much of what comes in, unless it does something, it's going to get lost. Ultimately, whether it's going to be an advantage to get rid of it, or whether there's nothing to keep it there, it'll get lost. And so you have kind of rapid turnover. And so they were able to estimate about 1/3 of the genome in E. coli has turned over since it's separated from Salmonella 200 million years ago, somewhere in that kind of timeline. That sounds like a lot. Is it enough to actually kind of see this difference? And it turns out it is. When they sequenced 3 E. coli, they were able to finally kind of make a Venn diagram of what is kind of in common amongst these 3 E. coli. And they took a very conservative approach. This was just simply genes that were at all similar to each other. Doesn't matter where it's 99% or 95 or 90. I mean, I think they went down to like 70% or something. It was quite low. And just ask, of the genes that are even present, how many of them are shared between the different bugs? And it turns out, of these 3 bugs, only 40% of the total genes from these 3 were shared amongst all the E. coli. And so this differs rather dramatically from kind of the picture of, like humans versus chimps, where we essentially have all of the same genes. We have different alleles at most of them, but they're relatively few, probably new genes that have been introduced in either lineage since that period of time. But there's nucleotide variation, I don't know the right number, 95%, 98%, I'm not sure exactly what the number is. Here, all within E. coli, we'd all call these E. coli, only about half of each bug's genome is even the same as the other bugs. And then there are different bits that they kind of share, pairwise or unique to each other. And so not all bugs have been quite as dramatic as this, but I think one of the biggest and most surprising things that has come from the fact that we now have about 1,000 microbial genomes available, and there's at least a couple thousand on the way, is just how extensive horizontal transfer has been. And makes it suddenly very interesting and challenging in terms of trying to define, right, what is a species and what makes, we did call these all E. coli, so something in this intersection defines it somehow as E. coli, but yet this is our little lab strain, K12, this is a uropathogen, and this is the jack-in-the-box bug. And so something else, either in terms of new genes that are present uniquely, or in terms of alleles, that they have different versions of the same gene, certainly there can be plenty of nucleotide differences in this, also then defines why they're all kind of different from each other. So I'm going to turn back away from horizontal transfer, but as you'll see, I'm using it as the system for my experiments. So, like I said, in the lab, we do experimental evolution of populations of bacteria. We use Methylobacterium, not quite as large and puffy as this one here. So there are many different ways. I mean, you can culture bugs however you want when you do experimental evolution. There are various advantages to using kind of different sorts of techniques. I'd say by far the easiest is to simply do kind of serial transfer, where you inoculate a flask of fresh media, you let it grow up, and then you take a small amount, you inoculate a new flask, you let it grow up, etc. And so, often people ask me, so what are you selecting for? I'm not, green eyes or something new. I'm not selecting for anything. Essentially, it's natural selection. Whoever can make more cells than other genotypes will become, more represented in the population. And so, even though again that the environment is completely and totally artificial, they never evolved in flasks at 30 degrees in my incubator, in this media mix with only one carbon source to eat, and there's no phage attacking them, there's no other bacteria around, they're not on a plant anymore. I mean, everything is different, you could say. But whatever does go on here in this new world, this is natural selection. They can either become faster, they can find ways to feed each other, they can find ways to kill each other. It really doesn't matter. That all of these things can contribute to their fitness, how fecund they are. And so we do this for a period of time. We have a population size, so these transfers are every couple days, 64-fold dilution, so this is 6 doublings for each transfer. And we have a final population size of about 2 billion in 10 milliliters. So again, this is a very large number compared to thinking of packs of cheetahs. And thus selection is by far what we're looking at here relative to drift. And so I have an incubator like this that's stacked up so it doesn't take up a lot of room in my lab because we grow a lot of cultures. And kind of the most important aspect, I suppose, about experimental evolution and using bacteria is that you can save them alive in the freezer. So I have the ancestor alive in my -80. I have the entire population at a series of times. We can plate out, take isolates, save them, and look at them. So we have this living fossil record of what's occurred, and we don't just do this once. We have replicates for any given situation, and then we have different environments or different genotypes that we can start out with. And so the great thing is you can do the experiments, and then long after, when new techniques come around, like I'll talk about some of the sequencing techniques that are available now to find mutations, new ideas come up, etc., they're still there. And so you can go back, dig them out, and do the experiments kind of after the fact, which is fantastically useful. And so I wanted to just briefly mention, in terms of getting at this point, in terms of how bacteria grow. If you look at some number of cells with time, these curves, as probably most of you know, look something like this, in terms of cells, and then thus the substrate goes, something like that. Does that look believable? So this is exponential growth with a carrying capacity that I don't artificially set. It's just because there's only so much carbon substrate that I add in there. And so this is if you just simply plot the number of cells. It's much more useful to plot the log of cells, because then you get something that looks like, well, except for they don't quite line up, but where this exponential process becomes a perfect straight line if you plot it as a log, of course. And then from this you can have a period where this is exponential growth. You have typically this initial period of time here where there's a growth lag, where it takes them this is somewhat idealized. They don't just in an instant suddenly achieve maximal growth. But essentially, there's a period of time where it takes them to kind of, we all have a lag time, right? When we wake up, same thing here. It takes them a period of time to physiologically adjust to a new condition. And then there's stationary phase. For most of us, this is maybe about 2 p.m. But this is where they've exhausted their food, and they're in a period of starvation, waiting for the next day's resources. And then in our case, we transfer them again, and they go forward again. And so most of the selection, it depends on kind of how much you dilute them and how long the lag phase is and whether they die in stationary phase. If there's a really long lag, or if once they finish growing, they die really rapidly, those can be aspects that that selection can act upon too. But assuming they don't die really quickly and that your lag is relatively short, almost all of the selection is upon kind of the maximal growth rate of one cell versus another. And so if you do experimental evolution, then what do you end up seeing in terms of kind of how fitness changes with time? And I think it's really going back to this kind of macro microbe. These can look really different from each other. So people do selection, of course. They do selection with macrobes. You've got people in Wisconsin and cows, and you bring together the best bull and the best cow that produce the nicest milk or the best meat, or corn with the best oil amount, or things like that. And so when we tend to think about selection with macrobes, we tend to select upon a diverse population, and or people do this in the lab also with fruit flies. They may take they have a colony of 100 or 200 fruit flies, not a billion, 100 or 200, of some mixture of genetics, and then they'll select for tolerance to ethanol or whatever. And in these situations, if you look at fitness with time, any guesses for what this looks? So you start some arbitrary place in fitness. Does this go just a straight line forever, really fast and slow down, slow and then speed up? If those are your three choices. And everyone has to vote. Any takers? We've got a C. We've got a lot of C's. We've got an A. Nobody's going to go with B. All right. Well, that's a good idea. Yeah, so with macrobes, actually it tends to be A, because initially you have a lot of variation in your population. And so what's essentially occurring each generation, and typically you do sample each generation, because this could be years or something like that, depends. You're winnowing down that variation, and you have a small population, so there tends not to be that there's almost no new mutations because there are such small populations. What you're really doing is you're selecting upon new combinations. This is assuming that you don't have migration from the outside or something else like that. But so in the beginning, when you've got a lot to potentially work with, the new combinations, your F1 hybrids, your F2s, your F3s, et cetera, there's a potential for some part of that kind of leading edge in whatever phenotype you're kind of, typically in these cases, they could be artificial or natural selection, but that might be relatively far out there. But once you've kind of made your best kind of combination of alleles, you're basically stuck. And this will actually flatline until someday in such a small population you get some new mutation that actually is better. But these populations just tend to be very, very small. For microbes, I'll warn you ahead of time, this is actually a trick question. So it's a trick question because it's actually kind of, to some extent, it's actually a combination of two of these. Initially, it looks like C. and where you don't see anything happen for a little while. And so why is that? The reason is I'm seeding these populations with one genotype. Initially, there is no variation. If there's no variation, your population cannot improve. And so you get new mutations. And first, so no variation at the start. So you have a new mutation that arises. In how many cells did it arise? One. And what's our population size? Approximately 1 billion. And so it's a very small proportion. And first, we transfer only 1/64 of the population. So it has to be lucky enough to end up in my pipette tip that first day, at least one of its progeny, and then again and again. So for a little while, it just has to escape drift. And so many good things will actually get lost just at the beginning because they were unlucky. It could be the best thing that ever happened. And just, if I don't sample it that first day, well, it basically didn't happen. If it does escape drift, once it's enough of the population so that these 64th-fold dilutions don't lose it, so maybe 3, 4, 500, there's no way you're going to end up with 0 in the next transfer, then it's kind of this relatively kind of methodical rise, just like compound interest. It'll just slowly rise up through the population, but it's still starting at 10 to the minus 7th in terms of proportion. And so there's a long waiting period before things happen. If I plot this on a log scale, it would actually be kind of just rising up at a straight line. But it'll take a long time before you see the population start to move. So initially, it looks like this. And this would be until, how long does it take to see something happen? In my experiments, it actually only took about like 60 generations. We already saw a lot. Sometimes it can be much longer. But this is, somewhere on the, usually this is, tens to hundreds in terms of kind of the timeline to begin to see something happen. It depends. If you put a selective pressure that's going to kill them, you'll see it essentially in the first generation because nobody's growing. And so if you put them on antibiotics, then that's different. But so if you go in the long term, and so the lab I was a postdoc in Michigan State, Rich Lenski, maybe some of you have heard of it. He's run a long-term experimental evolution with E. coli and minimal glucose. And so they've run an experiment like what I've done, but it's run for nearly 50,000 generations, so since like February 15th, 1988. And so every day, in this case, transferring and going through about 6.7 generations. So if you look in the long term, this is the answer. So essentially, the composite is there's a little bit of a lag in the beginning, but then on the long term, adaptation tends to be relatively quick early on and tends to slow down but not grind to a halt later on. And this is for many, many different systems and many environments with different bugs. This is almost always, essentially always been the case, with very few exceptions where suddenly something dramatically different emerges. And actually, in one of those lines, something dramatically different did emerge. But if you're curious later, I can tell you more about that. But in any case, over the long term, kind of the way of reasoning this, and actually it does have to do with the work as a whole, is that initially there can be some kind of low-hanging fruit. There are some very kind of large things that you can do to improve the situation in some new environment. But as you begin to fix the easy things, it's not that there's nothing you can do, but everything at some point, there's kind of some intuition, and which tends to play out that after a while, there's not much you can do to become 50% better. There may be lots of things you can do to become 2 or 3% better, but these things tend to become exhausted, and so you do continue to improve. It's just that the pace in terms of fitness tends to slow down. So these are kind of the picture of what we tend to see when we do experimental evolution in the lab. So the nice thing is when we do this work in the lab, we can actually assay fitness. And so like I said, fitness is relative reproductive success. This is how we're defining it. And the nice thing is we don't, if you look at plants or something like that, you could think of fitness as being like, how many berries does this bush have, or how many eggs did this bird lay, or something like that. And first, that could be kind of just very stochastic from one year to the other. Next, birds could have different numbers of eggs or whatnot. And it's not to say, are 4 eggs from really healthy birds the same as 4 eggs from really unhealthy or bad moms or whatever, kind of thing. It's kind of a component of fitness, how many children that you have, but it's not everything. Also, how good are the children themselves, et cetera. And so the nice thing is, again, in the laboratory, we can kind of integrate over the entire environment and measure the relative fitness of 2 bugs by doing a competition, where we put 2 in together. And the way we do this, evolved strain, we can take our ancestor. In this case, we've fluorescently labeled it. We put in a version of GFP that's yellow. It's called Venus, brightest thing in the sky. That's why they named it that. You can grow them up each separately in whatever environment you want to test them. Usually, this will be the exact same environment you did the selection in. You could do others. You could ask, if you select on methanol, do they become worse at other compounds? And we have a whole line of work about looking at that sort of stuff. But you can mix them together after having grown them separately, kind of get them physiologically acclimated. And then while they're growing together, you can do one or you can do more cycles of growth where you go through this entire growth phase with both being present. And you can then look at how things change from the initial time point to the final time point, and you can count them. Now, this used to be, in my postdoc, this was sitting there with a little clicker and counting how many pink colonies and how many white colonies there were. And this is exceedingly painful, and you get bad data because you can only count so many colonies on a plate, so there's tons of statistical, just numerical fluctuation if you're counting 30 of one type and 100 of the other. Whereas if we use a flow cytometer located in the basement of the Bower, it counts 50,000 events in about 40 seconds. And so you get much, much, much better. I mean, there's hardly any experimental noise, really. And the nice thing also, you're following a ratio. And so, I mean, this is really, so many things end up being controlled for. And so you can look kind of initially, we can gate in terms of those cells that are fluorescent and the majority of cells that are not, and see how this changes in time. And so, like in this case, the non-fluorescent cells that were the evolved type, increased in number substantially relative to the fluorescent cells, the labeled ancestor decrease. And from this change, we can then calculate kind of a proportional fitness differential per generation. And so we'll report a fitness value of like 1.2. And that would mean that per generation, I'm 20% more fecund than you are. And so, I would leave 20% more offspring per generation. And so then that number will, depend on how many, it's just like compound interest, how many generations you let that run in terms of how it affects the proportions then of the two types. So the nice thing is we can measure this very easily, and it, like I said, it captures kind of all aspects of the growth curve. And so there are some caveats involved here, but it's a very nice and effective way to actually be able to measure adaptation as it proceeds. And so, what's the system? So there's kind of two ways of addressing this question. I've kind of actually outlined both of them. We wanted to look at it, try to be able to kind of predict adaptation before it happens. And to do this, we need a system that we can kind of think about and model, et cetera. We also have to know that whatever kind of subsystem, I don't want to model the whole cell. I can't do that. Right now, it's relatively unreasonable. And so we needed a situation where we kind of knew what is selection acting upon, that it's not, not all the genes are relevant, but just kind of some of them are relevant. So something that a particular module of how the cell works is under selection. And also then this module that we're working with is one that we horizontally transferred in. And so it relates to kind of this other aspect in terms of horizontal gene transfer. I wanted to address how, when you do get new pathways that come in that allow you to become a pathogen, that allow you to use a new substrate or whatever, how did these become incorporated into the rest of the genome and the rest of what the cell does, and that the cell didn't do that before. Those genes weren't present in this bug before. You've kind of got this new matchup between these two different parts. And so those are kind of the set of questions for which this work was trying to address. And so I took my bug, Methylobacterium, that grows on methanol, and I took its particular pathway. I could draw in much more detail if you wanted, but trust me, this is nicer, for how it gets from formaldehyde to formate. And if you remove this pathway, if they can't deal with the formaldehyde, I don't think you even need to say your guess. In fact, the slightest fume of methanol stops them from being able to grow when they're on other compounds that they could normally grow on. And so it's quite toxic if you get rid of this red pathway. However, in a strain where I got rid of that pathway, if I put in now a foreign pathway, and so I'm doing the transfer, it wasn't transduction, I guess it was conjugation in terms of how I introduced it, but I made the plasmid that buries a foreign pathway. This foreign pathway gets this from formaldehyde to formate, but they're completely different enzymes that are not related at all. They use different cofactors, different chemistry. It's a different way. It'd be like ripping out your gas engine and putting in an electrical engine. And my guess is your car today is probably not ready to really run very well if we did this. For example, your battery is probably way too small to be able to handle such a change, et cetera. And so the nice thing is the cells do grow by having put in this foreign pathway, but they grow threefold slower than the original. And so they're alive. And so like this is actually an important aspect, that if you put in some foreign pathway, you need to have something to select upon in the beginning, particularly if you're going to do this in the lab, so that you can see things improve in time. But the fact that they're specifically slower just because of kind of this one change, it's not a trivial change, but there's kind of this one specific change from one pathway to another pathway, it led me to speculate that this is what selection is likely to act upon. And so, I mean, the issue has been in previous work, so like the E. coli for now almost 50,000 generations, it's been really, really hard to possibly, even after the fact, rationalize the mutations that have occurred, because they've just occurred in all sorts of various physiological processes. Penicillin binding proteins, which have to do with cell wall biosynthesis, topoisomerases, which have to do with coiling up the DNA, and they're just all sorts of things where, I told you that they're growing on glucose. So you'd think all you have to do is turn to your big map of glucose metabolism and say, it'd be this gene, then that gene, then that gene. It's hard. We don't have kind of a complete and quantitative understanding of cells. And so what's nice about this system, I think, is that this replacement hopefully will kind of winnow in kind of the low-hanging fruit to being this particular pathway that we know how to think about well, we can measure things about, and we have a model for. So that the initial largest effect beneficial mutations, which will be the first to rise up in a population, will be something that has to do kind of with this part of the system. And so I'm going to skip the aspect in terms of Michaelis-Menten kinetics and just mention that so we can model this system, which really looks, and even this is simplified, something more like this. Luckily, we don't have to model this. I mean, this is basically what I'm trying to say, is that the hypothesis is that the flux through kind of this initial bit from methanol till you get to biomass, my hope is I only have to worry about kind of a little chunk of the total map and not everything. It's not to say that everything else doesn't matter, but I hope that that matters a lot less than the part that we can think about. That's the basic idea. And thus, we can model these each of these various reactions using MATLAB and using differential equations. And so, this is Michaelis-Menten kinetics, where you can describe each enzyme in terms of a Vmax and a Km, et cetera. Except they get ugly if they have multiple substrates and reversible and stuff like that, which is why some of these just look really hairy. But in any case, this allows us to describe, how the system should be working, as best as we understand it. And this system, it's important, two things. Number one, even though this is kinetic, so it changes through time, we end up modeling the change in all the various concentrations or fluxes through time. It actually reaches a steady state, which is essentially equivalent to exponential growth rate. That as long as methanol is around, it ends up coming to a steady state where it's growing at the same rate until methanol finally goes away and then it slows down. And so it does kind of achieve something, it's that steady state growth rate that we're trying to be able to match up how the model predictions to how the bug's actually growing. And in collaboration, so I have a collaborator at BU, Daniel Segre, who's fantastic, and he's a modeler. I'm an experimentalist, but we know enough about each other's fields to be able to very well communicate with each other. And so he's developed a genetic algorithm to help fit some of the parameters. Luckily, even though, so this model has about 80 parameters, which is quite a lot, in terms of all the Vmaxes and Kms, et cetera. Luckily, we actually have published values for about 60 of them. And so we have at least a starting place for approximately how active is this enzyme or this enzyme or this enzyme, how well do they bind to this, et cetera. And so that gives us kind of somewhere to begin kind of our search. So the question, all right, great, this model, what do we want to do with this? Well, first I'll just say, so we can, in terms of the fitting, we can ask it to, try to predict the relative fluxes through the various different pathways, kind of how much goes down each road, how fast the wild type cell grows, how fast this kind of new cell grows, et cetera. So there's actually, even though there's tons of parameters, there actually are lots of different types of data that that need to be fitted, which is important if you want to try to make something like this work. Why do we do this? Is we're trying to use this model to address optimality, in that we're assuming that we're trying to ask if we could change. This gets back to that kind of that first question in the 4-step program. If you could change anything, which change would be the most effective? And so using this model, we're able to ask, if I could bump up any enzyme's Vmax, if I could change any enzyme's Km, how much effect, proportional effect, would it have flux going into biomass and thus how fast they grow, and thus how fast they over-reproduce the other types. And so, you can, if you look at how these sorts of predictions look, for many enzymes, if you look at, for example, Vmax, is you increase Vmax if you have some system of enzymes where they're all needed, and you have some flux going through them. These curves tend to be something like this, kind of quasi-hyperbolic type curves. And for many enzymes, actually, quantitative variation in their levels has relatively little effect in kind of flow through the system. You need it. As you begin, ultimately, if you dropped it too low, things will start to slow down. But there tend to be relatively few enzymes for which, at their current level, if you quantitatively varied it a little bit, would have a kind of a large effect on flux. And in fact, so you can describe every single enzyme as having some slope at its current point, and this is its quote control coefficient, how much it could change flux if you could change its amount. And kind of one neat, there's an area called metabolic control analysis that describes this, is that for any system, even the entire cell, the sum of these control coefficients is always 1. And so there's only, you can't, like every single part in the cell cannot have absolute control over what's going on. It's not unlike driving the car. There's only one steering wheel. Usually one person has control over it. Sometimes someone in the back seat has some control over it too. There's only so much control over the system. And thus it's distributed over many enzymes. And so the average, if you're talking about the whole cell, the average control is very, very low. If you have 500 enzymes working to grow on glucose, something like that, then the average is going to be very, very small. But it's not to say that you aren't going to have at least some that have larger effects and many more that have just very, very, very small. And so that's the whole point of this model, is to figure out which of those that have the larger effects and by how much. And thus, these then provide quantitative predictions in terms of how much selection is acting upon those enzymes. So you're kind of in one step. These models allow us to bridge from kind of understanding how systems work to understanding how evolution can act upon them. Because if we know what parts we could change to go faster, this is the hypothesis, is that then this is where we'll find beneficial mutations. And so thus we can make our control curves for all the various parts in our system and asking which of these are steep and which of them are completely flat. And in terms of thinking about adaptation then, something like this is a horrible target. We're at least predicting that there's basically nothing I can do to an enzyme that looks like this that will possibly make the cell grow but infinitesimally faster, whereas there are some aspects that in terms of the predictions of the model at least, you could get a lot of effect from having changed those parts. And so I'm happy to talk about it. I don't want to burden people with a lot of results, and I know I've probably gone through a substantial amount already. I'll just summarize a couple things. First is that the adaptation has actually been very fast, and so in terms of those kind of curves, I don't have enough time points for you to actually see. These actually do look like this, where there was a period where they kind of sped up and changed, et cetera. But we see rapid adaptation, that even though they sucked when they first had this pathway, they got better much faster than, the rate of adaptation was actually pretty phenomenal compared to like E. coli and glucose. These guys have doubled in like 120 generations, whereas the E. coli lines still have not doubled on average after 50,000. They're closer, about like 80%, something like that. But obviously, this isn't fair. These guys were kind of in a fitness well. They were not doing very well to start, and so the fact that they can improve proportionally a lot isn't that surprising. And then like growth rate and flux through the system, as well as fitness, all correlate well. So this aspect that you can kind of relate the models, the flux that you predict, to the growth rate, to fitness, tends to work out well. And their improvement has been actually very specific to methanol, the C1 compound that goes through this pathway. When we test them on something else, succinate, a 4-carbon compound, they haven't really improved in any consistent pattern at all. So in this case, they're not just better to being in our lab and going through our cycles. Most of the improvement is actually very specific to the compound that goes through this pathway. So let me just say, I'll summarize in terms of, then what actually happened underneath the hood. We found a number of mutations through kind of traditional approaches. One was pretty, if you had any candidate to start, it'd be the pathway you put in. It's certainly a good place to be thinking. It's very easy to think about. And sure enough, in every single population, the pathway we put in had beneficial mutations all across the board. They all had the same effect. I'm going to zip through some of this. In a wide variety of places, this is the plasmid that bears the foreign pathway, which is encoded by those two genes. And in all cases, these beneficial enzymes actually reduced the enzyme activities by twofold. And this led to an 18% growth increase. So the other thing I forgot to mention is kind of an advantage of working with microbes is, besides measure this increase, once you find the genes that have mutations, you want to ask if they're beneficial, you can move them back into the ancestral background. So you can have the exact same ancestor that now only has this one mutation present. It can be a single nucleotide difference, and ask how much effect. Do the same sort of fitness competition that we could do with some evolved strain that had 10 or 15 changes. We can do it with ones that contain just one at a time. And so what's interesting here is that there appears to be an intermediate optimum. And actually, here is a case where the model fails miserably, because in this case, less enzyme is more. And so in terms of thinking about the model, all of those curves that I showed you, they were all monotonically increasing. How could less be more? The thing is, flux, even though it's a good way of beginning to think about a situation, it's not everything, because the fact that you're making the proteins in the first place potentially also imposes a cost, either in terms of the resources involved, or if they misfold and form kind of aggregates in the cell, they can have a burden also. And I put in, for this initial work at least, I put in this foreign system under the strongest promoter I know in the cell. And so they were like 8% of the total cell protein. And so we're working on it, but it looks like the reason why these, why less is more, is that the curve for benefit was flat, but the curve for cost just kept going up, up, up. And so there'll be some point, then intermediate, where you've reduced the cost, but not yet begun to reduce the benefit. And so you can get somewhat kind of more complicated examples like this, and we're working through them. And then in other work, we have found a number of other enzymes that relate to the system. And the one thing I want to just mention is, for some of these other enzymes, we're at least beginning to have reasonable accord between the model and the predictions. In that, for a particular enzyme that interconverts NADH and NADPH, which are needed for different roles in cell physiology, we have a particular beneficial mutation that cranks up the enzyme by 3.7-fold. We can measure that, and we can move that allele back into the old strain, and then we can measure how much effect does it have on the strain, and we can ask our model, exactly, not just is it good, but how good would it be if you could increase this by 3.7-fold. And so the model prediction was an 8% increase, and based on this curve, and our experimental data was a 12%. So now, of course, 8 and 12 are not the same value. But if you consider most of the predictions, the vast majority of predictions would have actually been like 0.001, 0.001%. This is actually reasonable. This is beginning to sound like the model has, at least for some of these features, is fairly robust. And we've had another case where we had a beneficial mutation that gave a 44% increase in growth. This is a fairly substantial effect in terms of what's been typically seen. And we don't yet know, we know it affects the amount of a particular cofactor that's made in the cell, and we're trying now to measure just how much it went up so we can make a quantitative prediction. But the neat thing is this is the biggest selective coefficient we've seen, and it's in the most sensitive parameter of the model. So at the very least, in terms of kind of like rank ordering what we've seen and what the model predicts, this is also very comforting that the model is at least in the direction of being able to make some of these predictions in terms of what would be the sites in this bug's physiology where beneficial mutations are likely to occur. The very last thing I wanted to leave with is just kind of where this type of stuff is going. So I think the hardest part in my mind is the sort of stuff that I've already been talking about in terms of what do you do with mutations once you've found them. But for a long time, the hard part has been how do you find the mutations in the first place? I mean, so much of genetics has been about breaking cells. If you get some cell that can't grow on some compound, you can isolate mutants that don't grow on this, or isolate cells, put some gene's promoter behind put GFP behind it, look for cells that suddenly glow when they shouldn't be glowing, and go find something that's really discrete and obvious that you can see as a colony on a plate, that kind of phenotype. These phenotypes, this is growth in liquid that's being selected upon, and a 3 or 5% change is huge in terms of evolution. You will beat your neighbors quite handily if you're 3 or 5% better than them. But this is not something that you can find easily. It's not going to jump at you on a plate in the same way. And so, how you find these mutations has been a challenge for a while. And one reason why we're only now kind of being able to use these sorts of systems, not only to ask evolutionary questions, but to ask kind of these more physiological questions. And so the neat thing is, now we just sequence them. And so like the Human Genome Sequencing Project, there are ways of doing sequencing that have become remarkably cheap. There's one, so we collaborate with the Broad Institute down over by MIT, the shared Harvard-MIT Institute. And there's a particular new technology called Solexa, and it generates many, very, very, very short reads, and which you then use to try to assemble a genome. And so it can generate 5 million 36 base pair reads, and this costs $1,500, which is very, very cheap compared to anything before. So the quality along these reads actually goes down with time, but a student of mine ran like every single genome that was in the database and found you actually need, basically the question is, if you only have 36 base pairs at a time, it would be like 36 letters of the Bible, and then you try to read, that have been shredded up many times over, and you try to put the damn thing back together again. I probably shouldn't use that word in Bible in the same sentence, does it actually work? And he found that actually it only takes about 19 or 20 nucleotides before you can get to basically being able to find essentially every single position uniquely. It doesn't take very much. And so these short reads actually do a fantastic job, at least with small genomes that don't have too many repetitive elements in terms of being able to resequence. And so, for example, with 15x coverage, we can capture about 97% of the genome, and the remainder is just repetitive elements, things that occur more in one place. And thus, the data is something that looks like this, where this is the genome sequence of the ancestor at the top, and here's a C. And these are all a series of reads, little short reads, that can align, that include this position, and that they've been kind of ordered so that they look nice and pretty. And you can see every single new read coming out has a T, whereas it was supposed to have been a C. And so this, very easily we can identify new point mutational changes. And so this is being used in cancer genetics to do exactly the sorts of things I was talking about before in terms of identifying the variety of mutations that are involved in cancer. It's being used in, to look at HIV populations, look at populations of Pseudomonas and cystic fibrosis lungs, et cetera. It's a very nice technology, and it's very appropriately used for trying to to find de novo small changes that might be hard to find otherwise in populations like this. And so from like one focal isolate that we looked at, we found 2 single nucleotide polymorphisms like this. We've seen 2 small indels, like a deletion of 2 nucleotides and one, I think, of 11. One insertion sequence that hopped, a transposon, a mobile element that hopped into upstream of a gene. 2 large deletions, one of which incorporates 10% of the genome. And there were several endogenous plasmids. One of those is now gone missing. And so from this technique, you can actually get a very wide variety of changes. It's really pretty fantastic. And frankly, I mean, $1,500, it's not that bad. So I'm developing a teaching lab here at Harvard. And so we're actually going to sequence, as one of the various projects that students are going to do, we will sequence one of the isolates we've already evolved in lab, and the students can then compile this data, find the mutations that they think are present based on the data, and then go after, determine are they actually present, and then if they are present, move those alleles into the correct genetic systems to be able to introduce them back to the original strain and measure their effects and what other phenotypes we see emerge, et cetera. I mean, it's something that actually, I think this sort of data is amenable, at the very least, to kind of a longer-term laboratory course. It may be difficult to try to squeeze in, I mean, to carry all these sorts of things through, but I actually think it wouldn't be too hard to think of. Some of these programs are not so bad. We're actually developing some nice software that we hope to have like a nice graphical user interface to be able to look at this stuff. And so the ability to actually take some data like this and have it be assembled and look for new mutations, I actually think that would be something that would be on the relatively feasible side of things now. It's quite nice. So I will leave with that. I won't talk about the work in terms of epistasis, in terms of how various mutations interact with each other. And I'll just mention, Like I said, a lot of this work has been in collaboration with a colleague at BU who's fantastic, Daniel Segre, a number of other collaborators, notably also those at the Broad Institute who have been doing this sequencing for us, and people who give us money. And then this is my lab who does the work.